Subnautica gave its largest creatures a classification: Leviathan-class organisms. Players have spent years building community wikis, estimating their sizes, arguing the numbers in forums. The creatures are fictional. The numbers are extravagant. What those wikis rarely mention is that 3 real animals, alive today, would sit comfortably in the same category, and that nobody has fully explained why any of them grew that large. 3 of them are what this is about. What you're looking at is real footage from NOAA Ocean Exploration's 2017 Gulf of Mexico expedition, a United States federal scientific agency, archived and citable. The animal on screen is Bathynomus giganteus, a giant isopod, approximately 30 centimeters in this particular clip, with confirmed specimens reaching around 50 centimeters. It's a deep-sea relative of the pill bug in your garden, scaled up to the size of a rugby ball and filmed by a remotely operated vehicle at depth. Beside it, a scientific illustration drawn from specimens recovered between 3,890 and 8,931 meters. Alicella gigantea, the supergiant amphipod. Maximum confirmed body length, 340 millimeters, roughly the size of a loaf of bread. A scavenging crustacean found across roughly 59% of the world's ocean floor, according to a 2025 global distribution study in Royal Society Open Science. And then, museum specimen footage from Te Papa Tongarewa in Wellington, New Zealand. A mature female Mesonicoteuthis hamiltoni, the colossal squid, caught in February 2007 in Antarctica's Ross Sea. Total length, post-thaw and fully extended, 4.2 meters. Weight, approximately 470 kilograms by Te Papa's refined estimate. The heaviest known invertebrate on Earth. 3 animals, 3 verified sizes, 3 separate sets of questions about why any of them ended up this way. Start with the isopod, because it has the most evidence. Bathynomus giganteus was first described by the French zoologist Alphonse Milne Edwards in 1879, from specimens pulled up near the Gulf of Mexico. The genus name means roughly deep sea family. The species name speaks for itself. For more than a century, specimens turned up occasionally in trawl nets and bait traps. Pale, compound-eyed, armadillo-shaped, and far larger than anything in their order seem to have any clear reason to be. The maximum verified body length sits at approximately 50 centimeters, established in a 2024 Zookis paper citing Lowry and Dempsey's 2006 taxonomic revision. Popular media has circulated a figure of 76 centimeters for years. That number does not appear in peer-reviewed taxonomic literature. It appears to originate from early press coverage that predated careful measurement. The 50-centimeter figure is the defensible one. Most B. giganteus live between 365 and 730 meters deep. Over 80% of confirmed specimens have been collected in that band. The species can appear as shallow as 150 meters and as deep as around 2,140 meters. They prefer mud or clay seafloor, and the way they eat isn't inferred from captive behavior. It's confirmed from gut contents of wild animals. Briones Forzan and Lozano Alvarez published the first systematic stomach content analysis in 1991, examining 158 specimens caught off the Yucatan Peninsula. Fish and squid remains dominated. Barradas Ortiz and colleagues followed in 2003 with the first study tracking diet across life stages. Fish and cephalopod material remained primary at every phase. Galatide crabs, echinoderms, and occasionally other isopods, including conspecifics, also appear in documented stomach contents. The diet table drawn from these peer-reviewed studies lists named squid species, Loligopialei, Rossia, alongside fish fragments and smaller crustaceans. They aren't selective. They are also frequently not well fed. The stomach of Bathynomus jamesi, a closely related giant isopod species, occupies roughly 2/3 of the entire body cavity, far larger than that of their shallower water or intertidal relatives. A finding documented in the research context surrounding the 2026 Cell paper. A 2022 BMC Biology genome paper by Yuan and colleagues at China's Institute of Oceanology describes expanded gene families linked to thyroid and insulin hormone signaling, efficient nutrient absorption, and a low basal metabolic rate. The paper's language is efficient mechanism of nutrient absorption, storage, and utilization. Then, in August 2026, Yuan's group published a follow-on study in Cell documenting what they call a dual adaptive strategy. A food-retentive stomach enabling episodic hyperphagia, gorging when food appears, combined with a markedly depressed baseline metabolism. The Cell paper also identifies a gene called ND1, apparently acquired millions of years ago via horizontal gene transfer from microbial lineages, which appears to reduce the body's baseline energy requirements during food-scarce periods. The result? Giant isopods can go more than 5 years without eating. A captive male reportedly went over 1,500 days without food. That's a captive observation, not a wild measurement, but the genome data gives it biological grounding. The energy conservation hypothesis, the proposal that cold, food-limited deep environments select for suppressed metabolic rates, efficient nutrient storage, and extended growth periods, is the leading proposed mechanism for isopod gigantism. Crustaceans have indeterminate growth, meaning they keep growing throughout their lives rather than reaching a fixed adult size. If metabolism is deeply suppressed and growth continues slowly over a long lifespan, bodies of 50 centimeters become achievable without requiring continuous large food inputs. A reduced predation argument also circulates. Fewer active predators at depth may release size from certain constraints. That idea has intuitive appeal. No formal predator list for adult B. giganteus has been compiled in the peer-reviewed literature. The absence of documented predators isn't the same as no predators. What remains genuinely unresolved? The genome work was done on B. jamesi, not B. giganteus itself, so it's not confirmed that the same expanded pathways appear in the most widely documented giant. Wild growth rates haven't been directly measured. Whether the energy conservation mechanism causes the size, or is an adaptation that developed alongside it, or both, the current data can't yet separate those options. Now the amphipod, and here the species identity matters because it's frequently reported incorrectly. The animal is Alicella gigantea, not Eurythanes gryllus, a different deep-sea amphipod found globally, including at hadal depths. Not the amphipods of Challenger Deep, which include Hirondelia gigas, a separate species, not this one. Alicella gigantea is the sole member of its genus, described originally by Chevreul in 1899, and it's the world's largest confirmed amphipod, reaching 340 millimeters total body length. Harrison and colleagues established that figure in 1983. Barnard and Ingram confirmed it in 1986. Jameson and colleagues reaffirmed it in a 2013 paper documenting the first in situ filming and recovery in the Kermadec Trench. The 2025 Royal Society Open Science Synthesis by Moroney, Niazi, and Jameson, compiling 195 records from 75 sites worldwide, cites the same 340 mm maximum. A common shrimp is around 5 centimeters. Alicella gigantea is roughly 7 of them end to end. Its range runs from 3,890 to 8,931 meters. It occupies the lower abyssal and upper hadal zones, the places where pressure is measured in hundreds of atmospheres and sunlight has been absent for geological time. The 2025 study concludes that despite being observed infrequently, A. gigantea likely occupies roughly 59% of the world's total ocean area by depth range. It isn't rare. It has been undersampled. What it eats has been observed rather than systematically dissected. Alicella gigantea belongs to the Lysianassoid line of amphipods, a group broadly recognized as carrion scavengers. In situ observations from baited landers confirm they aggregate at fish bait at around 6,700 meters in the Murray fracture zone. Jameson's 2013 Kermadec Trench paper documented them entering baited traps at 6,265 to 7,000 meters. Fatty acid and stable isotope analysis from the New Britain Trench suggests a dietary shift from microbial and planktonic debris in juveniles to carrion in adults. There has been no dedicated stomach content study for this species comparable to the 1991 Briones-Forzan work on the isopod. Diet for A. gigantea is inferred from trap bait feeding observations, taxonomic affiliation, and chemical proxies. That is a meaningful evidentiary gap. The food scarcity paradox is acute here. A 34-centimeter body requires sustained energy to build and maintain. The deep environments A. gigantea occupies receive organic material mainly as slow particulate rain and occasional large carrion falls. How the animal manages this metabolic arithmetic isn't yet resolved. The 2025 distribution paper notes low observed population densities, though suggests this likely reflects sampling limitations rather than genuine rarity. That observation doesn't resolve the paradox so much as reframe it. The mechanistic evidence specific to A. gigantea comes from a 2021 Frontiers in Marine Science paper by Lee and colleagues, comparing whole-body transcriptomes of A. gigantea from the New Britain Trench against the smaller amphipod Bathynomus schellenbergi. The analysis identified genes evolving faster in A. gigantea that are linked to growth regulation, glycerolipid metabolism, starvation response, and meiosis. Lee's team explicitly state that temperature and oxygen concentration alone are insufficient explanations. Altered growth regulation genetics and starvation resistance strategies are their proposed drivers, not Bergman-style environmental determinism, but molecular-level selection acting on specific gene pathways. A 2017 genome-size study found that A. gigantea has the largest genome among the deep-sea amphipods examined, around 34 picograms. There is a correlation between genome size and maximum body length across the species examined. Correlation isn't mechanism, and the authors don't claim otherwise. What remains unresolved, the causal chain from altered growth regulation genes to 34-centimeter body, hasn't been closed experimentally. The food paradox, large body, sparse food, has no resolved answer. Whether Challenger Deep contains A. gigantea, as frequently stated in popular accounts, isn't corroborated by peer-reviewed distribution data. The apparent conflation with other hadal amphipods is long-standing and should be resisted. The colossal squid is the hardest case to get right, partly because of a different animal that gets in the way. Mesonica teuthis hamiltoni isn't the giant squid. Architeuthis dux, the giant squid, is a separate species with a global distribution. The largest confirmed giant squid measured close to 13 meters total length. The colossal squid's confirmed total length is 4.2 meters. It's shorter. But it is heavier by a substantial margin, roughly 470 kilograms against the giant squid's documented maximum of around 275 kilograms, with a denser body, a larger and more robust beak, and rotating hooks on its arms rather than suckers. Different animal, shorter, heavier, and more powerful per unit length. The reference specimen is the one caught in February 2007 by the New Zealand longline vessel San Aspiring, owned by Sanford Limited, while fishing for Antarctic toothfish in the Ross Sea. The squid was incidental. It came up on the line, mature female, publicly announced on 22nd of February 2007, donated to Te Papa Tongarewa by the New Zealand Ministry of Fisheries. Post-thaw, fully extended, 4.2 meters total length, 2.5 meters mantle length, approximately 470 kilograms by the museum's considered estimate. Initial news coverage the day of the announcement described the animal as 8 to 10 meters long, deckside estimates, taken before post-thaw measurement. The 4.2 meter figure is what the ruler produced. Te Papa describes the specimen as the world's largest preserved invertebrate. It currently sits in a tank in Wellington, and you can visit it. Adults are inferred to reach 2,000 meters or more, based on sperm whale dive depths and the recovery of colossal squid beaks from whale stomachs. That deeper limit is inference from predator diet, not direct observation. What the colossal squid eats has been established partly the same way, backwards, through what eats it and what analysis of stomach contents and isotopes can reconstruct. Specimen-based stomach content studies suggest fish including lanternfish and Patagonian toothfish, other squid, and possibly conspecifics. Documented depredation events on Antarctic toothfish longlines confirm active predation on large prey. Now, the metabolic rate, which is the most striking single number in this animal's biology. Rosa published a metabolic modeling paper in 2010 in the Journal of the Marine Biological Association of the United Kingdom. This isn't the same as Rosa and Cittable's 2010 paper, which concerns Humboldt squid, a different species entirely. Rosa's colossal squid paper estimated a mass-specific routine metabolic rate of 0.036 micromoles of oxygen per hour per gram at 1.58 degrees Celsius, the approximate temperature of the squid's Antarctic operating environment. Projected daily energy consumption, roughly 45 kilocalories. For a 500 kilogram animal, this implies a requirement of approximately 0.03 kilograms of toothfish per day. A single 5 kilogram toothfish at 80% assimilation efficiency could sustain such an animal for around 200 days. The colossal squid uses almost no energy at rest. Rosa describes it as likely an ambush predator, patient, slow-moving, conserving everything it consumes. Its metabolic suppression appears to exceed what cold water temperature alone would predict for comparable squid. The proposed defense hypothesis adds a separate proposed mechanism. A 2009 Current Biology paper by Nilsson and colleagues argued that the colossal squid's eyes, the largest of any living animal, around 27 to 30 centimeters in diameter, evolved specifically to detect the bioluminescent signatures of approaching sperm whales at depth, giving the squid enough warning to escape. The paper extends this to suggest that whale predation pressure may have driven body size alongside eye size. Larger body, better escape odds, stronger selection advantage against a primary predator. A 2012 analysis by McLean challenged whether the eyes are specifically tuned for whale detection or simply reflect a general allometric scaling relationship. The body gigantism as defense argument has one substantive supporting paper and one substantive challenge. It hasn't been settled. What remains unresolved? The 4.2-meter confirmed length almost certainly does not represent the species maximum, given that beak sizes recovered from sperm whale stomachs suggest larger animals exist. Reproductive biology and wild growth rates are nearly completely unknown. Whether body gigantism in M. hamiltoni primarily reflects metabolic emergent properties of living cold and slow, or reflects specific selection imposed by sperm whale predation, or both operating at different timescales, the data currently can't separate those explanations. Across all three cases, one pattern recurs. Large bodies, environments where food arrives sporadically and in small quantities, and metabolic strategies built around doing very little for very long periods. That profile broadly fits the energy conservation hypothesis. Cold water slows metabolism. Indeterminate or extended growth proceeds slowly across long lifespans, and large bodies become reachable without requiring sustained high food intake. The theoretical framing goes back to Timofeyev's 2001 review in the Biology Bulletin of the Russian Academy of Sciences, which proposed that deep-water gigantism and Bergmann's principle share a common mechanism. Colder habitats produce larger cells, longer lifespans, and extended growth periods. Timofeyev's paper is a synthesis of existing data, not an experimental test. Applied to ectothermic marine invertebrates, the Bergmann-type pattern has documented support in some groups. Certain copepods and amphipod families follow the trend, and contradictory results in others. Craig McLean's 2006 Ecology Letters paper found that deep-sea gastropods follow an island rule pattern instead. Small-bodied, shallow-water ancestors grow larger at depth. Large-bodied ancestors shrink. No single rule governs all of them. The 2022 isopod genome paper and the 2026 cell paper from Yuan's group provide the most mechanistically specific evidence so far. Direct genomic and biochemical evidence that B. gemesi has adapted its molecular machinery for metabolic suppression, possibly through horizontal gene transfer from microbial sources. That is a much more specific claim than cold makes things large, and it points towards species-specific molecular evolution rather than a universal environmental rule. Which brings the question back to each animal individually. For the giant isopod, does energy conservation best explain the size, or has the evidence simply not found a predator defense argument worth testing? For Alicella gigantea, is growth regulation genetics the driver? Or is there a food processing efficiency story waiting for a dedicated gut content study to reveal it? For the colossal squid, is 470 kilograms the product of an extremely slow metabolic pace sustained over a long life? Or is there a 10 million year arms race with sperm whales buried somewhere in the body size data? The science hasn't closed on any of those. Radically different environments can require radically different adaptations. That much appears to hold. But which adaptation explains each case, and whether one mechanism ever dominates cleanly over the others, remains the open question the researchers who know these species best are still working through. The dataset here is 3 animals. The deep ocean is mostly unmapped. Whatever comes up next in a baited trap or an ROV clip may not fit any of these 3 patterns at all. Subscribe if you want to follow where the evidence goes.